All right, so, oops. Oh, yeah. Right, so hello everyone, and yeah, welcome to the Biostats seminar. So today we are very pleased to have Dr. Jean Morrison, who is an assistant professor of biostatistics at University of Michigan, which she joined this year after completing a postdoc in the Department of Genetics at the University of Chicago and a PhD in our department. So Jean's research is primarily motivated by problems in statistical genetics and genomics, and her current focus is on the analysis of high-dimensional or structural phenotypes and causal inference. Jean has interest in empirical and variational Bayesian methods and genomic applications of deep learning. So a quick reminder to the attendees to put your questions in the Q&A box or to raise your hand to be unmuted and pose the question yourself if you prefer. And so yeah, we're ready to start. Jean, thank you for accepting our invitation. And yeah, we look forward to your talk.
Thanks, and thanks for inviting me, and I will share. Okay, hi everybody. I'm really excited to be speaking here, so thank you very much for the invitation. So I thought I would just start with an introduction, and you already introduced me, so you're just going to hear the same material a little bit twice, but I don't think I know a lot of the people who are probably on. So I wanted to say hi. This is a picture of not Michigan in the winter, but also a lovely place. So just, I guess, my CV information that you already told you. I got my PhD here at the University of Washington with NOAA in 2016. And then I did 4 years as a postdoc at the University of Chicago in the Department of Human Genetics, which was very cool because I got to, interact with people who were sort of all along the spectrum of very statistics to, very interested in biology and developing new assays and sort of work on a variety of problems along that spectrum. And sort of while I was there, I got interested in causal inference a little bit, which I'm actually not going to talk to you about in this talk. I'm going to talk about something else. And then just 3 months ago, I joined the University of Michigan biostatistics as an assistant professor. So I'm just sort of getting my feet wet here and figuring out how this all works as the world continues to be in a sort of odd place. So research-wise, I have a sort of small collection of questions that I've been interested in and sort of poking around, and they're all sort of in the space of statistical genetics or human genetics, having to do with the relationship between the human genome and human trait variation. And these sort of range around just can we figure out what variants are causing what traits and through what mechanisms? And then also, can I use what we know about the way that variants work to make causal claims about other variables? And then I think also a really interesting question or avenue is, can we use variants to predict traits or drug responses or disease progression, that sort of thing. So I'm going to talk to you about today is a project that I've been thinking about for just a little bit, and it sort of it kind of halos around a few of those topics that I just talked about. So at the heart of this research is sort of what can we do with data that we've gotten out of genome-wide association studies. So for a minute, I'm just going to pause and tell you what a genome-wide association study is. If you're already in statistical genetics, this is extremely boring and you've heard this a lot. But if you're not, it's important to sort of know what this foundational study in genetics looks like. So in a genome-wide association study, we take a group of people And classically, we measure them for one trait that we might be interested in. And then we also measure their DNA at a bunch of places. So that gives me out, capital N measurements of a phenotype Y. And then for each person, I have J, where J is very large, different variants that I've measured. And then the thing that classically people do with this data is they just run capital J linear regressions and estimate marginal effects. They estimate the marginal effect of xj on the outcome y, and we call these summary statistics. So what this ends up looking like, if I take the summary statistics for a single trait, so here I've taken coronary atherosclerosis, people often make this kind of plot where they plot the position on the genome, and then on the y-axis they plot the association between the variant and the trait that they're particularly interested in, as they usually do negative log 10 p-value. This is called a Manhattan plot because it looks like a city. So this particular trait that I pulled out, coronary atherosclerosis, we can see that there's lots of peaks, and that happens because variants are actually correlated with each other. Variants that are very close to each other in physical genome space will be correlated in the population because of how DNA segregates. So we get these sort of peaks of association. And this has been what people have been doing for a long time. And in fact, people have been doing it for so long that we now have thousands and thousands of these studies. So you can also sort of like flip the axes that you think about. And instead of thinking about, I want to look along the entire genome at a particular trait, you can think, all right, I have this one variant that I want to look at its associations over. My internet is on that impacts this in some way. So I have this one variant, and I'm going to look at it over lots and lots of traits, because now people have done lots and lots of GWAS. So in this case, I pulled out this variant that is, it is associated with coronary atherosclerosis. So this plot is sort of analogous to the Manhattan plot. The vertical axis now is traits, though, instead of positions on the genome. The y-axis is still negative log 10 p-value. And these traits all happen to be ICD-10 codes, which are disease diagnoses that might end up in an electronic health record. So this variant is associated with coronary atherosclerosis. It's also associated with gallstones, and I'm just going to say gallstones because I had to have Google play this word for me twice, and I'm still not confident that I can say it. It's also associated with hyperlipidemia. So this variant is like potentially doing a bunch of things. And that sort of gives us a bigger picture of what's going on than just to say, okay, this variant is associated with heart disease. There's a little bit more happening here. So this phenomenon where a single variant is associated with many traits is called pleiotropy. And the idea of pleiotropy has really driven a lot of my research in recent years, and been sort of inspirational for how I've been thinking about associations in the genome. So the phenome, as opposed to the genome, so the phenome is like the collection of all of the traits that we've studied by GWAS. The phenome is getting extremely large. So this is a database that I actually really love that's been put together by this group in the MRC IEU. So this database has like 34,000 GWAS in it. So you can just sort of go there and say, I'm interested in all of the associations with eating red meat, which is in there. You can go look it up. Somebody's looked up associations between variants and eating red meat. You can go look it up. So there's just thousands and thousands of these in here, which gives us kind of an amazing opportunity to start to get some context about the activity of these variants. I'm no longer stuck with just, I know this is associated with heart disease. I can go look and see all of the other traits that it either is or is not associated with. So a lot of people have been thinking about pleiotropy for a while, and there's a few approaches that you could take. A lot of people sort of started with just thinking about like pairwise relationships between traits, which like makes sense. That's I think, the like clear way to start. So one way that you can think about the pairwise relationship between two traits in terms of their genetic associations is genetic correlation. So genetic correlation, we're trying to estimate the correlation of the genetic component of two traits. You can think about like partitioning a trait into its like genetic component and its environmental component. If I knew what the genetic component of the trait was for lots of people, what would be the correlation there? So that's like a single number that summarizes sort of a whole bunch of things about the genetic sharing of two traits. Sort of the opposite end of that scale is co-localization. So instead of thinking about the entire genome in aggregate, co-localization is thinking about single variants. So it will take, a very small region of the genome and try to figure out, okay, we know there's a variant that's associated with both traits in this region. Can I figure out, is it actually causal for both traits, or is it like two traits that are one causal for each, two variants, one causal for each trait, and they're correlated, and so it looks like they're both affecting both traits. So that's co-localization. Mendelian randomization is sort of like an oddball also approach. In that case, we're interested in actually asking the question, is one of these traits causal for the other one? And that sort of creates its own particular pattern of variant association. So there's all of these ways of like thinking about pairs of traits together. The other option is, instead of thinking about traits two at a time, just like look at all of them. So think about pleiotropy sort of across the phenome. So I made this plot when I got sort of like really interested in HDL cholesterol and like what variants are associated with HDL cholesterol and what else are those variants doing? And, why does HDL cholesterol seem to be associated with some things and not with other things? So that's sort of a whole different research story, but I'll just sort of pare this down to what this plot is. So I took all of the variants that I could find that were associated with HDL cholesterol, and then I pruned them for LD. So remember I told you that variants in the genome are correlated. I said, I want to just look at independent ones. So I pruned down my set of associated with HDL variants to just a set that seemed to be approximately independent from each other. And then I went and I looked up all of the other traits that those variants were associated with. So in this plot, what I've done, one row of the plot is a variant. One column of the plot is a trait that I got out of that big database of GWAS. And I've colored a square green if the variant is probably, I think up and down doesn't matter, but let's say the variant is increasing the trait, and I've colored it purple if the variant is decreasing the trait. And I just oriented all of the variants so that they're all increasing HDL. So we can sort of read this and we can say, okay, this variant that is increasing HDL, it's decreasing, whatever this trait is. Then I grouped the traits and I grouped the variants so that we can sort of see some patterns. So along the horizontal axis, right, I have different sets of traits. First, I have HDL on the left. So you can see they're basically all green because that's what I selected these variants on. And then there's like sort of different components of HDL. So there's like very large HDL and like medium-sized HDL. That's this like middle green block. And then there's lots of traits that have to do with LDL cholesterol and different sizes of LDL cholesterol. We get into red blood cell traits like hematocrit and various aspects of like how many and what size of red blood cells do you have. There's some white blood cell traits. How many like granulocytes do you have? There's a ton of traits that have to do with adiposity or body size, things like waist to hip ratio, fat mass, that sort of thing. And then at the end, there's these diabetes traits. And I've left off a bunch of other sort of like sporadic traits. As vitamin D came up and blood pressure came up. But I think something that's interesting about looking at this plot is you can see that there are variants that appear to be doing really similar things. They appear to have really similar patterns, like across this entire set of traits. By the way, this is, I just sort of simplified it into a binary trait. So it's either yes or no. in terms of associated with the trait, and I set a threshold. So that's sort of why the plot looks, simpler than you might expect at first. So yeah, so you can sort of notice these patterns, right? Of there's variants that have very similar patterns across traits. It's not missing data. So if the variant just wasn't measured in a study, that square is colored gray, and there's almost no missing data on this particular plot due to what I chose to show you. So those white squares are genuinely white squares. So this sort of leaves me with the question, okay, it makes sense that variants would act sort of together, right? Because maybe you have two variants that are doing a very similar thing. They're like affecting the same pathway. They're, both of these variants are, changing the level of a particular lipid fraction or something, and then that's going to have a similar outcome. So can I identify these groups of variants that have a similar profile? And then if I can identify them, and I can identify that pattern that those variants are associated with, is that biologically informative? My hypothesis or my supposition is that will be biologically informative. It will tell us something about, the major like modes of action in the body, or the major pathways that can affect change. So why would this be interesting to do? It tells you a lot of things. It can tell you, if I alter this pathway, what side effects would I expect to see? If I'm acting on something that I know this particular variant is changing, and I know that these variants also change, all of these other traits, I might be concerned about some side effects from that particular medication. It's also, I think, useful just for like basic biological discovery, figuring out like how do things work and why do people get diseases. Sort of comes down to like what are the physiological pathways that are being perturbed, and this is sort of one way of like viewing physiological pathways. So this problem of can I find the variants that have a shared mode of action, we can sort of like translate back into statistics or math land as a matrix factorization problem. So I've sort of drawn the cartoon of what the matrix factorization looks like on the left, and then on the right, I'll write out the math, and you can think of it sort of either as the picture or as the math, like whichever, comes more naturally to you. But on the picture side, so we can imagine that there's this set of capital K unobserved factors. We've measured our large number of traits that are in the phenome. We have the genome worth of variants. And then we can imagine that these variants are acting through a smaller set of factors, and these factors have particular set of effects on the traits. So I won't hold us to literally interpreting this model, but it's sort of helpful to imagine that this is how it works for a minute as we set the model up. So if this is what's going on, then I can write each trait just as a sum of the effects of each of its factors, and then plus an error. Here, I'm imagining that this is maybe for a particular individual I, but I didn't want to subscript all of these things with I, so we'll just add that in your head. So I can then take these factors, which are hidden, and I'm just going to write the factor as the sum of its genetic effect. So we'll suppose that these are purely genetic factors, although they might have some errors, and I'm just not showing that here. So if I translate these ZKs into the sum of their genetic effects, and then I rearrange some terms, I can see that I now have a formula for the association of variant J and trait M, which I'm going to call beta J. So that association is just the sum over all of the factors of the factor effects times the sum of all of the variant effects. So that if you've seen matrix multiplication before, you're looking at this and you're saying that's a matrix multiplication, and indeed it is a matrix multiplication. So if I put all of the variance into this matrix that's capital J by capital M columns, then I can write this formula that I've written out as just LF transpose, where L is a matrix of the variant effects on factors, and F is a matrix of the factor effects on traits. So of course, this model is restrictive. The idea that all of the effects of a single variant on a particular trait would be mediated through a small number of common factors is maybe unrealistic. So we can add in some additional effects. So each variant can have like an additional effect that's just on Y, and it's not shared by any of the other variants. And you can imagine here arrows going from each variant to each trait, but that would be too many arrows. So I didn't put them in, but there's just one here for an example. So we'll call those effects theta. So now our matrix multiplication problem, we have capital B equals LF transpose plus theta. By the way, feel free to jump in and ask questions. I can't tell if people have questions, so just feel free to interrupt me. So finally, why am I writing this all out? Because really what I care about is the patterns in the association estimates, which are what I can get my hands on. Generally, I don't have access to all of the individual level data, but I have these databases and databases of summary statistics, so I want to use that. So if I have effect estimates, then I have this additional estimation error term. I can add the estimation error term to my my matrix representation of this formula. So I just want to take a minute and say, so these factors, the ZK, I sort of presented them like they might be real biological factors, and they very well might be, but in fact what they are in this model is they are convenient linear combinations of traits. So they're linear combinations of traits that represent the major modes of action of variance across traits. So that's sort of another way to think about them, because I think that, you know, in the reality of the body, which is much more complicated than this sort of 3-component model that I've shown, we don't really think that all of the effects are going to be linear. And in fact, whatever these hidden factors are, they're almost certainly connected to each other in some complicated network. So this is sort of a convenient way of visualizing and characterizing the major modes of pleiotropic action of genes in the genome. So just like another like visual representation of what am I doing when I say B equals LF transpose plus theta plus E, what does this matrix factorization actually look like? So on the left here, we have our B hat, which is that matrix of effect estimates that I got, you know, out of the database or from somewhere. L has one column for each of those hidden factors. So it has capital K columns, and it has one row for each variant. F has also K columns and then M rows, and then it's transposed. So in its transposed form, it has K rows and M columns. So we can see for this like top variant, it only is loading on, and so I'll maybe like alternate between saying loading and then like affecting, because I think loading is more accurate, and then affecting is the like cartoon mental model. So this variant is loading on this very last factor. So if we think about, okay, what is its effect going to be on each of these traits, I can just go to the last factor and I can say, okay, this variant is running the last factor program, and that's what its association is going to look like. And then we have this, you know, extra matrix of both estimation error, and then we'll say like sporadic or like independent other effects. So that's sort of this is the model that we're thinking about. I've suggestively in this cartoon made L and F sparse, so they have a lot of white space, which means they have a lot of zeros in them. And the reason that I did that is I think it's either convenient or, you know, related to true biology to make our models sparse. And the reason that we do that is because I think for one, it's nice to just have a smaller number of things to think about, to say like, okay, this pathway is about lipids and that's it, instead of this pathway is about lipids and a little bit about everything else in the genome. That's like harder to understand. And then I think it does correspond to biology in some way to say, all right, there are a small number of big, large effects, and I want to focus on those, and I'm going to shrink everything else to zero. So that's sort of what we're thinking about when we say this is sparse. We're thinking about, okay, each of these hidden factors, there's only a small number of variants that load on each of those factors. And each factor is only going to actually be contributing to a small subset of traits. And that's going to be what makes these factors interpretable. Okay, so now I've translated my biological problem into a matrix factorization problem. Great. How am I going to fit it? So there are a lot of matrix factorization approaches. There are many, many ways that you can go about estimating L and F depending on what assumptions you want to make. And I have not even come close to getting all of them on this slide. So don't get mad at me if I left off your favorite one. So probably the most common and like popular matrix factorization method is the SVD. So yeah, we all know and love the SVD. And if you don't know and love the SVD yet, you will in the future know and love the SVD. So what is the SVD doing? It is assuming that those factors are orthogonal and dense. SVD, if you don't truncate it, will give you as many factors as you have the rank of your matrix, but you can truncate it and say, just give me the first few most explanatory factors. You can take the SVD and make it sparse, so that's convenient. There's non-negative versions. You can assume that every entry in one or both of the matrices is positive. And then there's sort of grade of membership or topic models where you assume that all of the entries in a particular row sum to 1. And then there's tons and tons of other ways to go about this. So what are we going to do? I'll pause for a minute and go back and say, I think one thing that's good to like wrap your head around about these matrix decompositions is this decomposition into L and F is not unique. So it's only unique up to, I can reorder all of the factors and I can get exactly the same answer out if I reorder L and F simultaneously. And then I can also flip signs. So I can flip the sign of a particular row and then flip the sign of a particular column and flip the sign of the corresponding row on the other side and also get the same answer out. So often and then scaling, right? We can scale as long as we scale symmetrically, we can scale rows and columns and get out the same answer. So usually for convenience, we'll represent, if we've estimated these L and Fs, we'll represent them by just scaling everything in a particular column for L or a particular row for F transpose, a column of F. We'll scale it all to have norm 1. And then just remember that the sign of the relative sign, within a factor matters, but the relative sign across factors is not unique and you can change those orders. So that's something that's just like useful to remember. Okay, so what are we going to do for this summary stats problem? So there's another of these. So there's a whole set of Bayesian methods for decomposing matrices that I didn't even put on the previous slide. So one of them is this method called empirical Bayes matrix factorization. So this is something that Matthew Stevens was my postdoc advisor. They were working on this method a couple years ago when I was getting started. And it has some really interesting properties. So I may alternatively call it eBMF and Flash. Flash is the name of the software. So those are inconveniently like different names. So those are the same thing. So eBMF takes each of those columns of L and each of the columns of F, and it says, okay, I think each column is from some prior distribution. I'm not going to tell you which one. I think each of the columns of F is from some prior distribution. And then I'll say that the elements of E are independent. We're just going to you can say that either we're ignoring theta or like wrapping it into E, depending on how we estimate tau. So what am I going to do about the prior distributions? Well, I'm going to say the prior distributions are from a family that has some like friendly properties. So it's either like a point normal, or it's a mixture of normals, it's a mixture of uniforms. You can do a lot of things. And then the plan is going to be, I'm going to estimate the prior for each of these columns, and then I'm going to estimate the columns and the rows, columns and rows. So I'm not going to go into how to fit this. I think it's pretty interesting. It uses a variational approximation and an iterative approach. But I'll leave that to the paper, which you can go and read about how to estimate this. So this has, I think, a nice property that the sparsity of the factors is learned from the data. So that's one of the downsides of the existing sparse methods, is you have to sort of tune the sparsity of them, either using cross-validation or by putting in some information that you think you know about the data. It also, as implemented, is able to select the number of factors, which is a really nice feature for us. So it'll generate sparse factors that actually like conveniently also are not orthogonal. So orthogonal means that if I multiply the two vectors, I get I get 0. Sorry. So there's sort of no reason that we would think that like underlying biological factors are orthogonal. So not making that imposition is maybe convenient. It also maybe makes the space hard to fit. So that's, you know, trade-offs. So I thought, okay, maybe using this eBMF method is gonna work out. I didn't a priori say like I'm, you know, committed to using the eBMF method for this. I just wanted to like get some interesting information about what these variants were doing. So I start working on this problem and I realize, oh no, we've got this problem for the genetic data, which is that basically all of the matrix factorization methods assume that that E matrix, all of the elements of it are independent. And that's actually just not true for our summary statistics problem. So we have, I told you we can have correlation between the variants due to linkage disequilibrium, but we can get around correlation between the variants by just pruning. So the genome has, you know, you might get like 4 million or 12 million or 1 million variants measured in a particular GWAS. You probably like don't need all of those. You can just like prune away until you end up with a independent, approximately independent set of variants. I put a star by eliminate because you don't get down to no correlation, but we can say it's good enough. So correlation between columns of that B matrix, though, occur because many of the GWAS either are performed in the exact same sample or in overlapping samples. So we have these biobanks that have thousands of traits measured, and I have GWAS for all 1,000 of those traits, but they're all performed in the exact same set of people, which means that if there's environmental correlation, I can get correlation in the effect estimates that's just because of the environment, because it's all measured in the same people. So that gives me correlation in E, and so that's a problem. I have to deal with it somehow. So here's just a cartoon of what can happen when you have environmental correlation and all of the GWAS are measured in the same samples. So on the left, I'm going to show you some simulated data. On the left is the true genetic factors that I simulated the data with. There's 5 of them and there's 20 traits. On the right is the environmental correlation matrix. So this is, you know, it's obviously a cartoon. So it's got these like 5 blocks of 4 traits each that are all highly correlated and they're not correlated between. So pretty simple. So what happens if I just don't know that there's correlation in my errors and I just apply eBMF to this data? Well, I get out factors, but they're not the genetic factors that I was looking for. They look a lot more like the environmental correlation. So these estimated factors, if you sort of like look at this plot long enough to know what's going on. So in this matrix is is just like the blue and red matrix I showed previously. Each box shows the effect of one factor on one trait. So this, if I go to the true factors, this first factor, is having a strong effect on trait 20 and then an effect in the opposite direction on trait 4. So what am I estimating? I'm not estimating the truth. I'm estimating these like 4 blocks of correlated traits. So a lot of these factors look like the, I'll just spoil the answer. They look like the eigenvectors of our correlation matrix. And so that's not what we wanted. We wanted to find the genetic factors, but eBMF doesn't know. It just knows that you're looking for the patterns of correlation in the data. So this is what it gives you out. Okay, so what do I do about this? So if we're in the situation, so our problem is that the rows of E are correlated. I can get it so that the columns of E are independent by doing that LD pruning. If the rows of E are correlated, but I have this situation where I know the correlation structure, or I know the covariance of the row of E, I can do this trick where I take the SVD. So even though we're not using the SVD for the main problem, we'll still come back to it. It's still a good friend. So if I know the SVD, I can actually take my error matrix and I can write it out as the sum of E tilde, which is the kind of error matrix I wish I had. All of its elements are independent. And then this term that looks a lot like that LF transpose term, except I've put different letters in. So the V transpose, and I've used the superscript -m to mean I took off the last of the singular vectors. This also, by the way, that doesn't matter. So yeah, so the V transpose I know, so that came from my SVD. And then the A is random, but its elements are independent, which is convenient for me. And I know their distribution. They're all normally distributed, and their variance is the singular value. So this is the mth singular value from that SVD. So this looks like a factor decomposition problem, which is convenient because I'm using a factor decomposition method. So I can actually rewrite my model as B equals B hat equals LF transpose plus this new AV transpose term, and then theta, and then E tilde, which I like. So what do I have to do? I have to tell Flash, I have to tell the eBMF method, I'm going to give you these factors and I want you to fix them and don't change them. And I'm also going to tell you the distribution of A. And so that's fine. We can put all of that information in. And once we know that, we can then fit this whole model and get out an answer. So, all right, so that's nice. We don't actually have the same sigma for all J. And the reason for that is those beta hats don't all have the same variance. So the variance of the beta hat is proportionate to 1 over the variance of the SNP, or the genetic variant that you're looking at, times the square root of the sample size. So that can vary both across variants and then across traits. So if the sample size is constant across all variants, I can get around this by transforming my effect estimates into what I'm going to call standardized effect estimates, where I take the z-score and then I divide by the square root of the sample size. So that works if I have the same sample size for every variant, which is true sometimes. So sometimes, or approximately true. So, but a lot of times it's not true. And sometimes it's like wildly not true, because some of these studies will be like meta-analyses of lots of studies, and they'll just have wildly different sample sizes for each variant. So in that case, I can convert to z-scores. The factors for the z-score are going to be sort of different from the factors for the beta hats, but one convenient feature of them is their sparsity pattern will be the same, and generally the signs, because the signs of the entries will be the same. And then in practice, we find that estimating with z-scores versus estimating with standardized effects gives very similar results. And so we're not too worried about it. And what do I do about sigma? I just estimate sigma from some of the variants in the genome that really look like they're not associated with any of the traits that I'm looking at. Okay, so I do this in my toy example and I get back to some degree of accuracy the true genetic factors. So that's encouraging. I'll just remind you that so I reordered my estimated factors so that they would approximately match the order of what the truth. I didn't flip the signs, so I'll just remind you that these estimated matrices are not unique up to sign flips of columns. So this column is sort of a pretty good estimate of this column, even though the sign is flipped. So applying the fixed factor correction seems to work pretty well. I'm running a little bit out of time, so I'm not going to belabor the simulation results. So this simulation result is basically just showing that indeed correcting for environmental correlation helped us out. I'm going to skip that. Okay, so I want to talk a little bit about an application that I've been really interested in. So I primed you a little bit to be thinking about heart disease and lipids because I wanted to talk about this lipid example. So there's this sort of recent set of GWAS that have been put out for a large number of lipids and metabolites that were measured via NMR spectroscopy. And these are mostly lipids, features of lipids and amino acids. So I'm just going to tell you what is a lipoprotein because I don't know. And so this image is from Wikipedia, which is approximately my understanding of lipoproteins is the content of Wikipedia. So a lipoprotein is this particle that transports fats and hydrophobic molecules throughout your body. So your body is full of blood, which is mostly water. And so you need some way to get hydrophobic material from one place to another. So lipoproteins are basically this ball. They're hydrophilic on the outside and they're hydrophobic on the inside. And then on the inside, they can carry around these fats and triglycerides and cholesterols that need to get to different places. So when people quantify lipids in your blood, they're quantifying okay, how many of these particles do I find? What sizes of particles are there? So if you've heard high density lipoprotein and low density lipoprotein lipoproteins are very big and not very dense, as the name suggests. High density are small and dense, and they carry around different amounts of stuff. And in fact, it turns out that high and low is a very gross dichotomization of these things. There's all sorts of sizes and they do all sorts of things. So you can quantify a lot more granularly what's happening with these blood lipids through this NMR technique. And they've also measured some amino acids, but I'm not going to talk very much about the amino acids. So, all right, so I took these 123 metabolites. I made that matrix, that B hat matrix that was, millions of variants long by 123 traits wide, and I shoved it through our EBMF method with the sample overlap correction. And I'm just showing you the first 3 factors that I got out. So I've ordered these traits kind of to mirror some of the ordering that the folks who wrote that paper did, because I assumed they knew the good coherent ordering of the traits that would be informational. So this first factor, what's going on here, it's got this big block of green, a big block of effects that all go in the same direction. So in this plot, the vertical axis, each of these columns is a trait. And then on the horizontal axis, I'm just showing the direction of effect and the size of the effect of the factor that we're estimating. So this first factor is having a big sort of constant effect across all of the LDLs and very large LDLs. So these are the biggest particles. A variant affecting this factor then would increase the levels of all of the different VLDL features. The second factor is sort of the difference between the VLDLs and then the LDLs, and then I guess there's also IDLs, which are intermediate density lipoproteins. Can infer what that means. And then the third factor seems to be really about HDL, or high density lipoprotein. So this is just the first three factors that we got out. I thought that was interesting, but then I thought, like, okay, so then what? Like, what is that doing? One thing I noticed looking at this is the folks who wrote that paper really focused on the effect of something called LPA, which is also a blood lipid, but they didn't measure it in this study. LPA is known to be associated with heart disease. There's a gene called LPA, so that makes the protein LPA. There's a lot of name reuse here. So they focused on a particular variant in that gene, and because that variant is associated with heart disease, and it also has this like wide range of effects across lipids, and so they were looking for some information there about heart disease. So I looked at factor 2 and I said, that looks like the LPA effect that they published. It looks like the same profile. So is it really? And so I plotted it, and sort of. The answer is yeah, it sort of is. So on the vertical axis, I have the z-score for this variant that's in the LPA gene with a particular trait. And then on the vertical axis, I have the factor value for that trait. So for those strong effects, the LPA z-score is super highly correlated with the factor that we're estimating. For the other effects, the LDLs and the IDLs and the HDLs, it is still correlated, but it's less strongly correlated. So this factor is predicting sort of a strong association, whereas LPA is having a weaker association. And actually, what's going on with this variant is it's been assigned a loading to both factor 2, the one that I highlighted, and factor 1. And if you add these together, you actually end up just getting a factor that is only about very low density lipoprotein. So most of the effects of this LPA variant are on very low density lipoproteins. The other question I had, okay, so LPA is like significantly associated with factor 2. What about the other variants that are associated with factor 2? Are they also associated with heart disease? Is factor 2 like the heart disease factor? And that would be really exciting, right? So the answer to that is no. So if I look here, what I'm plotting, each point is a variant instead of a trait. So I'm plotting the association of the variant with factor 2, and I skipped the part where I tell you how we get the association between the variant and the factor, but I'm plotting the association of the variant and the factor, and then I'm plotting it against that association with coronary artery disease. And I've sized the bubbles by like the strength of their association with factor 2. So what we can see is some of the variants that are really strongly associated with factor 2 are really strong coronary artery disease variants, but a lot of them are not. So a lot of the factor 2 variants don't have anything to do with coronary artery disease. So factor 2 is not like a purely coronary artery disease factor. So it seems like there's a little bit more going on there than that we've like captured a single pathway. It seems like we've probably captured a like primary mode of pleiotropy, but there's like more granularity that we need to get at in order to get at like a single activity. So to that end, like it really seems like the thing to do is add more traits into the model. So add in coronary artery disease, see if that allows you to split apart that factor into multiple pieces, add in some of those like adiposity traits and some of the other traits that share pleiotropic effects with the variants that are important to this model. So I think that that's sort of the next step here, is to like enrich that matrix factorization to get like a more granular picture of the biology that's coming out of this. So in summary, the factor decomposition approach can reveal the dominant patterns of pleiotropy that are going on. Accounting for that sample overlap or the environmental correlation is critical for getting out genetic factors. And then biological interpretation, I think, is tricky, and it might like get better as we start to look at these like larger trait sets. So this is work that I started right before I left University of Chicago. So I've done it with Shin and Matthew, and then a couple of really talented graduate students at University of Chicago. And that's what I got. Thank you. I'm happy to take questions.
Thank you very much, Jean. Very exciting talk. We get a few questions. So the first one reads, is there any particular example of co-localization that is of interest in practice?
Oh my gosh.
The question was posed, I think, on the first part of the talk. We were talking about the 3 approaches.
Yeah, so there have been a lot of examples of co-localization. It comes up all the time. And so I don't have a particular example in mind, but it's often, it's something that people do often with gene expression. So you'll see a paper where they've taken, they have associations between their trait of interest and some variants, and then they have associations with gene expression that people try to co-localize like gene expression variants and their GWAS variants, and then they'll try to sort of conclude from that co-localization, like, okay, maybe this variant is affecting the trait through that gene expression. So that's not a specific example, but it's sort of that's how that gets used sometimes.
Thanks. And another question refers to the 2018 models by Wang Steven, I think, and is the attendee is wondering how certain priors lead to sparsity. So effectively, what kind of priors does the model use to induce sparsity?
Yeah, so typically, generally, all of the priors that we use with it have a point mass at 0. So it's like a mixture of a point mass at 0 and something else. So the point normal would be a point mass at 0, and then just a normal, so like the spike and slab model or something. And then you can have a mixture of normals. It's just like, that's like the spike and slab, except the slab is more complicated. So that's how you get the sparsity in there.
Thanks. And okay, so we have a question from Alan Wiseman, who says, I was pleased to see your treatment of correlation from shared samples in GWAS studies. I imagine it might be possible to extend the idea to truly shared correlation through similar unmeasured environments. Have you thought about how you might work that in?
I don't totally understand.
So it might be possible to extend the idea to truly shared correlation through similar unmeasured environments. I'm happy to unmute Ellen if.
Yeah, go ahead and unmute Ellen. And then, yeah.
Do I have a mic now?
Yeah, we can hear you.
Yeah, so, I mean, just think about, where we may have shared environment because of where we live.
Mm-hmm.
We might have independent people in our GWASs, but there may still be, cultural sharing or, other things that are shared that also affect the phenotype that aren't really measured.
Yeah, right? I see what you mean. So correlation that can extend like across, like, can there be correlation between effect estimates that are measured in different people, like because those people share an environment? Like, yeah. Yeah, I think there could be. And I think in this case, like, because we chose to use a method for estimating the environmental correlation that doesn't require us to know whether or not there is sample overlap, because actually it's sometimes hard to figure out.
It's really hard to figure that out, which is why what motivated my thought.
Especially if you're looking at like thousands of traits, you don't want to go in every single paper and then figure out like, okay, they used one of their cohorts is the same as the cohort that was used in like trait 465. So they have a sample overlap of 35 people, So that's really hard to figure out sometimes, and just like very labor intensive, even if the information is there. So we used a method that doesn't require us to know that. So I expect that we would be picking up some of that. I sort of glossed over some of the difficulty with that correction, which is that a lot of times the environmental correlation and the genetic correlation are very similarly structured.
Right.
So that makes, it's not impossible. There's some distributional differences that we assume. So we can still fit it, but it does make the problem hard. So I will say that. Yeah.
Yeah, I understand that.
Perfect. Tim Thornton raised his hand, so I'm gonna allow him to talk.
Hi, Jean. Can you hear me?
Hi, Tim. Yeah, I can hear you.
Yeah, very nice work. Very exciting. So I guess I'm thinking more about the potential issues with like not knowing the true causal variance. So, particularly like, let's say, for example, I'm trying to do this in a diverse population sample with population structure, and we really don't know what the true causal variant is, right? So for example, we know that variants identified to Europeans often aren't transferable across population. So what assumptions are we making about these effect sizes? And is it just restricted to, for example, European population, and you have pretty strong confidence that these are the causal variants? Is that one of the assumptions that we're making?
So I guess there's like two parts of that. So on the causal variant, like for a moment, let's assume that we, like all of our studies are in the same population for a minute, but I don't know what the causal variants are because it's still hard to know what the causal variants are. In that case, the pleiotropic patterns across variants are still informative if you assume that, what variance you're next to is going to be somewhat random. So if I don't think that like HDL and BMI variants are just always going to be next to each other, if I'm always seeing like a correlation between HDL and BMI, I can assume that some of those variants are doing both things. On the question of diverse populations, I think that's a really good point, and it's something that I had been working on a lot to try to figure out. Like, one of the things that we are sort of tacitly assuming is that the LD structures are the same between each of these populations. So even if what I've pulled out isn't a causal variant, it's a tagging variant, maybe it's going to be a tagging variant in all of these studies. And so if that's not true, or the genetic etiology of these traits is different in the different studies, which can happen even among study participants of the same ancestry, it can happen because they're in like different environments, then those interpretations get a little bit murkier. So that's, I think, that's something that I have not figured out how to address or how to, how to work with.
Okay, but we're saying that these are probably these are specific to a particular population. Is that kind of what we're?
They could be. So I think, and this was actually one of the reasons why I was like, okay, the first thing I'm going to look at, I'm just going to take a bunch of traits that are all from the same people, and then I don't have to like worry about those people being in different environments or having different ancestries or something. But I think the main issue that I'm thinking about is, a variant that might either it might do something different in one population versus the other, either because there's like an environmental effect or something else, or the LD structure might be different, so that what I've picked up as a tagging variant, say in population A, I have a tagging variant for HDL. Maybe if I'd managed to measure also, very low density LDL in that same population, I would get a correlation. But if I've measured it in a different population where it's not tagging that same causal variant, then I don't get that correlation. So I think that's the issue that I'm thinking about.
Yeah, that makes sense. Thank you.
So next question asks if so if the model can be extended to account for interactions between genetic and environmental factors, or interaction is partly addressed by the theta matrix.
Interactions between genetics and environment. I think that's interesting. I think you might need to have the same trait measured in multiple populations, which is something actually that I have thought about incorporating, because we can get that out of the databases. I can go and not just pull out, one GWAS for LDL cholesterol, but I can find GWAS for LDL cholesterol in multiple populations that have different environments. So I think there is a potential, I think, for doing something like computing the factors in one group and the other and then comparing. We sort of talked about, maybe computing factors in like men and women for particular like sexual dimorphic traits, and then seeing if those factors are different, and that potentially being interesting. And you could view that as like a gene environment interaction.
So in the model, how would it would be like multiplicative factor? Or
so that would be actually just like running two separate analyses and then comparing them is sort of what we thought about doing, rather than extending the model. There may be a way to extend the model and do it inside the estimation procedure. I'm not immediately seeing how to do that, but it might be possible.
Thank you. And I don't see any other question coming in. Perfect then. Thank you very much for joining us and for accepting our invitation. And thanks everyone for joining in. This is the end of the seminar. See you next week.
Thanks.